You've probably heard of cells, microscopic living units that compose everything that's alive, from humans to sharks, trees, fungi, and much more. Cells are crucial for us because they make our body function. They allow us to grow, gather energy, and keep our memories. Cells give us life as we know it. But how do cells know what to do? The answer is found in DNA. DNA is a giant recipe book that contains information on how to make any product the cells might need. The DNA's information is structured in chapters, the genes, and each gene holds information to create one product, a protein, the machine-like molecules that cells need to perform all their tasks. But how do cells make proteins? First, the cell finds itself in need of a specific protein. For example, if a cell wants to grow its cell skin, the membrane, it might need to build new proteins to incorporate and grow. To make these proteins, the cell first consults the DNA book to see which steps and materials it needs. The DNA book is kept in the nucleus, along with a set of specialized workers that search the DNA to find the right gene. These workers are RNA polymerases, molecular secretaries whose job is to photocopy the genes into much handier one-page copies, the mRNAs. This whole photocopying process is known as transcription, and it's a way to control how much protein is made, since more mRNA means more protein. The next step is to send the mRNA to be translated. This translation is done by a specialized machine, the ribosome, a molecular cook that follows the mRNA recipe to make the protein. mRNAs are written in three-letter words, the codon, and each codon matches with one of the 20 possible ingredients that compose the proteins, the amino acids. The ribosome's main job is to add the amino acids together in the order specified by the mRNA to make the protein. To do this, the ribosome receives help from molecular sous-chefs, the tRNAs. Each tRNA carries a specific amino acid and is capable of recognizing that amino acid's codon. So, when the ribosome reads a codon, it recruits the right tRNA, which then gives to the ribosome the amino acid to add to the protein being synthesized. This process repeats as the mRNA is read, until a specific codon is reached, the stop codon. The stop codon does exactly what its name suggests. It stops the ribosome from reading the mRNA, marking the end of the protein synthesis, which can now be used by the cell. Let's quickly revise the process. The cell wants a protein. The RNA polymerases search the DNA for the right gene, which they photocopy into mRNA. And then the mRNA is read by ribosomes and tRNAs, adding amino acids together to form the protein, until the stop codon is reached. Scientists discovered this process over 50 years ago, and since then they have been able to control how much protein a cell makes. To do it, they need to change the amount of times a gene is transcribed by RNA polymerases. And this is actually pretty easy to do. In front of every gene, there is a promoter region, which is essentially a neon sign that attracts RNA polymerases to copy the gene. By changing this promoter with a bigger, brighter neon sign, scientists trick RNA polymerases into photocopying the gene more, and therefore make more mRNAs for that protein. This is very useful because, as we saw before, controlling the amount of mRNA means controlling the amount of protein synthesized.
However, as synthetic biology evolves, the limitations of the transcriptional level regulation become more clear. Crosstalking, silencing, bursting, expression noise. So if we want to broaden the horizons of biology and meet the demands of the innovative research, we need something faster, smaller, and that allows control beyond the limits of DNA. We are introducing SKIPPIT, a first-in-class translation-level tool to control protein expression. By regulating protein expression as the protein is being assembled, we can achieve instant control of protein synthesis, regardless of previous layers of control. And to do this, we are unlocking the full power of RNA.
Recently, scientists have begun exploring how they could control the amount of protein made without affecting the number of mRNA created. To do that, they're trying to program the mRNA to edit itself, controlling protein synthesis during translation by making riboswitches. With riboswitches, the mRNA is able to scribble on itself to cross out or add parts to its own recipe, changing which amino acids are added to the protein or even if the ribosome can read the mRNA. Since their discovery, the research and creation of new riboswitches has been growing dramatically, and the vast majority of them control the ribosome. They work by capturing the region of the mRNA that the ribosome binds to, meaning that the ribosome can't read the mRNA.
RNA is a very complex molecule. It can suffer from multiple levels of processing, such as splicing or modifications, and it can even have catalytic or regulatory activity. A very interesting regulatory RNA are stop codon read-through elements. Usually, during translation, the ribosome reads a codon, adds the corresponding amino acid to the peptide chain, and ends translation once it reads the stop codon, releasing the peptide. In rare instances, there will be a stop codon read-through element, a naturally occurring untranslated sequence which interacts with the ribosome at the stop codon site, rewriting termination and allowing the ribosome to bypass the stop and skip it. The key to this element's function is its structure. Stop codon read-through elements are already very interesting on their own, but what if we could control them? And more importantly, could we manipulate their structure to turn it on and off? Aptamers are RNA elements that, when bound to specific ligand, adopt a particular structure. Therefore, by fusing the SCR element with an aptamer, we generate a fused element, where the SCR structure is maintained and its function is active, or on. Then, when we introduce the ligand, the read-through element binds to the aptamer and changes its conformation. This disrupts the SCR, which dismantles its functional structure, turning off. There is a simple way to explain the on and off states. In absence of the ligand, the aptamer's unbound conformation is preferred over its bound conformation. However, when the ligand is added, the binding energy of the ligand boosts the stability of the aptamer's bound conformation, making the bound conformation more stable than the unbound conformation. This difference in energies is what ensures a switch is truly capable of switching. And just like that, we've built a riboswitch. First, the functional RNA elements, which have either catalytic or regulatory activity depending on their structure, like read-through elements. And second, the conformational RNA elements, which have specific secondary structure that is altered due to an external stimulus, like an aptamer. Using our model, any functional RNA element can be combined with any conformational RNA element to produce next-generation RNA switches, opening a vast unexplored design space for regulatory RNAs.
But making RNA switches is not as easy as it seems. Despite RNA switches being extremely useful tools with a myriad of applications, without deep knowledge about structural and energetic design, it is nearly impossible to make them. That is why we created Tadpole, a novel software to automate RNA switch design. Tadpole uses our model as a conceptual core, which allows us to concentrate weeks of literature review and dry lab iterations into just one click. To generate a new riboswitch, a user just needs to input two things: a structure and sequence of both the conformational and functional RNA elements into our software. However, just stitching these two elements isn't enough to create a functional RNA switch. We're still missing one functional element: the linker. Linkers are short oligonucleotide sequences which allow the fusion of a conformational and a functional RNA element, allowing the user to exert better control over the conformation of the switch. After inputting our elements data into Tadpole, our software uses a wide library of random linkers to generate riboswitches. Each of these riboswitches is then evaluated according to their predicted structure and energy values. Finally, Tadpole will feed back to the user the most optimal riboswitches along with their respective linkers and predicted structures, and with an automated report of summarized results. We know that making RNA switches is not an easy task, which is why we decided to publish Tadpole to be available for everyone. With a public interface and a very easy-to-use website, we are opening the use of riboswitches to everyone, not just experts.
With SKIPPIT, we wanted to create something that has never been done before, a stop codon read-through riboswitch.
To do that, we first had to find a read-through element whose function was predominantly determined by the structure.
Our first candidate was a novel read-through element, SERD. As it has not been characterized, we set out to discover its functional architecture. We started by using Tadpole's multiple sequence alignment tool and identify key conserved regions in SERD's family. Then we generated constructs testing the variable regions and SERD on its own. What we discovered is that SERD is able to stimulate remarkably high read-through rates in hex cells, up to 9%, and that each of the different regions we tested achieved different read-through levels, generating a useful library of SCR elements with variable properties. However, as this element's function was not dependent on its structure, it was not ideal for riboswitch testing. That's why it was time for a turning point. That's when a well-characterized SCR element came into play, SKIPPIT. We tested it with the following device that contains a renilla luciferase, a firefly luciferase, a stop codon in the middle, and the SCR element downstream, which stimulates read-through. When read-through occurs, we get a stronger signal from firefly luciferase that we can plot against renilla and get the read-through percentage. With this powerful read-through element, we achieved up to 10% read-through in hex cells and a remarkable 12 to 14% in cell-free assays. This element became an excellent choice for our riboswitch design, as its function relies on its structure. We also successfully validated and optimized experimental conditions for the TO-on5 aptamer, which shows strong signal responsiveness in our cell-free system. Having both the read-through element and the aptamer, we were only missing one necessary final piece for the riboswitch construction, the linker.
We run the sequence of our functional RNA element, SEKIS, and our conformational RNA element, TO-on5, through our tadpole software. This tadpole software generated 4 riboswitches, each one with a different linker length, 5, 7, 8, and 9 nucleotides. According to RNA false structural predictions, SEKIS would be active in presence of the theophylline and inactive in its absence. Each of the candidate riboswitches has different free minimum energy values depending in its on and off states, with the energy gap depending in the linker length.
To be precise, we decided to test our 4 different predicted riboswitches, each with a different linker length. We observed a reproducible and consistent on-off switching behavior, with linker length modulating their switching amplitude. All 4 designs exhibited a clear ligand-dependent effect, with the L5 construct showing the strongest relative response. This shows that a shorter linker allows for a better design, and provides useful data to keep improving our computational tools. Together, these results mark the successful construction and validation of the first stop codon read-through switches. They also prove that our computational tools can effectively guide design. With all of this, we have built a library of SCR elements that can modulate translation at will. And we have also built a foundational platform for customizable riboswitches that other researchers can build upon.
But how far can we go? Riboswitches have slowly positioned themselves as one of the key fields of the future for synthetic biology, with a limitless amount of applications. Our SKIPPIT stop codon read-through riboswitch is especially interesting for dual protein expression systems, where a single transcript can regulate the expression of proteins upstream and downstream of the stop codon. This dual control can help the advancement of new synthetic biology fields, like biomanufacturing, industrial protein synthesis, and biosensing. Moreover, after thorough literature research and consultation with experts, we have seen how our innovative translational level control of termination could be applied to any field, from new therapeutics for cancer, to new cellular lines to study biological functions, to environmental applications, and so much more. With SKIPPIT, translational level control with RNA switches is only limited by creativity. However, sometimes limits are important. Misuse of our technology greatly concerns us. To guard against that, we decided to perform a thorough risk assessment and contingency plan. That worry reflects our deep commitment to our human practices approach. From day one, we engaged with stakeholders, collected feedback, and linked decisions transparently via HP cards through our wiki. And to go beyond academia, we delved into entrepreneurship with proposals to combine open source tools with protected IP, licensing SCR libraries, and offering Tadpole as a Saginaire engine to move forward market deployment.
And this comes at the perfect time for our university, as our scientists have developed a switchable protein system that is able to control human cells remotely.
Well, that is fake news. You may have noticed it, but not everyone else does. This is why we believe that truth and transparency should be cornerstone of all education. Without ethical education on science, there is no possible bright future.
A good scientist also needs ethics, creativity, good communicational skills, and an open mind towards new technologies. That's why the educational part of our project focuses on the many phases of science. That's why we created Reineta, a free and adaptable toolkit thought for students aged from 16 to 17. The main idea is to focus on a 360 scientist that not only masters their own area of research, but also thinks critically about the world around them. It's not only about experiments, but also about connecting science to society.
Each activity encourages students to develop essential skills rarely addressed in class. Debating an ethics dilemma, detecting fake news, exploring AI in biology, or linking art and evolution. To ensure our toolkit met the classroom's needs, we continuously gathered feedback from educators and experts through our HP work, shaping the future of Reineta. Even with limited means, we wanted to create something practical, evolving and lasting. And it's working. Teachers across Catalonia are already using Reineta, and it will soon be presented at a bioethics training program for high school educators organized by the Catalan government. Beyond the classroom, we have also taken part in events such as Festa de la Ciencia, the European Researchers' Night, and Catalunya Summit. We even translated the Arizona State iGEM children's book into our language and reached students online through a TikTok account with over 45,000 views. By bringing science closer to people, we are not just building knowledge, but also shaping a generation of future scientists capable of seeing the bigger picture.
RNA. RNA is the key to proteins. Control RNA and you control proteins. That is why developing new technologies that allow us to exert better control over RNA itself are crucial to move forward in synthetic biology. RNA switches are the key to unlocking the full potential of RNA, and we finally have the means to use them. From iGEM UB, we have developed the first ever recorded modulable approach to control translational termination using a read-through element. We are the first ever team to publish a read-through sequence to the iGEM registry and the first to characterize and generate protocols and devices to test them. We have also made a public accessible platform for anyone with or without expertise to be able to generate their own RNA switches. Everything we've built is more than a set of tools. They are building blocks, empowering researchers, students, and society to explore, create, and question responsibly. SKIPPIT shows that understanding and designing life at the molecular level is within reach, and with curiosity and ethics, we can turn the knowledge into a smarter and safer world. Thank you for coming along on this journey. Together, let's move forward, beyond the finish line.